My name is Hanjoong Jo. I'm professor of biomedical engineering at Georgia Tech and Emory University, also cardiology at Emory University. I am here to present our recent work on microRNAs, especially mechanosensitive microRNAs that is produced from endothelial cells, and how this microRNA regulates endothelial cell inflammation and atherosclerosis. So it's been well known that bad flow is associated with atherosclerosis. But it was unknown whether flow can actually directly induce atherosclerosis or not. So in 2009, we developed this animal model using mouse to demonstrate that if you change flow from good flow to bad flow by doing a partial carotid ligation surgery in mouse carotid arteries, we can then induce atherosclerosis within 2 weeks. So by causing this rapid atherosclerosis by just simply changing flow patterns, we were able to demonstrate that flow disturbance can actually cause endothelial dysfunction and atherosclerosis. So using this animal model, we have been studying how then disturbed flow causes atherosclerosis. So to further understand the endothelial gene change, we developed a new technique called endothelial enriched RNA preparation technique from these animals following the surgery. And this allowed us to do genome-wide studies, gene array, microRNA array, and DNA methylation studies. So from these, we were able to provide some really novel insights how at the gene levels, flow is really regulating endothelial function and how this then lead to atherosclerosis. Today's focus was on mechanosensitive microRNA, especially MIO712. We have shown that endothelial cells produce this MIO712 in response to bad flow in an in vivo situation as well as in vitro. And this MIO712 then targets one of the key protein known as TIMP3, or tissue inhibitor of MMPs or ATOMs. And so what happens is MIO712 goes up, this then directly downregulates TIMP3, which then activates MMP activity as well as ATOM activities. And by having these metalloproteinase activity really going haywire, endothelial cells become inflamed and they show increased permeability. This then leads to subsequent atherosclerosis development. We further showed that if we block this microRNA by using anti-microRNA 712, we can inhibit endothelial inflammation as well as atherosclerosis. Now we have further expanded this work to show that this anti-MIO712 can also inhibit abdominal aortic aneurysm as well. So we are now using this knowledge to translate into human studies by using some of the human homologs of this MIO712 that we identified from mouse studies. Thank you for listening.